Um, my name is Ahmed Hoque. I'm from Johns Hopkins Hospital, and I have Dr. Patrick Chinnery with me today, and we're going to talk about his research that he's presenting at the American Neurological Association meeting this year. Dr. Chinnery, can you tell me what prompted you to study mitochondrial mutations in HIV patients?
So the whole issue came to light over conversations with my colleagues in internal medicine who are interested in infectious diseases, and they described to me symptoms in patients that they couldn't explain that were becoming an increasing problem in their clinical practice. And it struck me that there was quite a similarity between the constellation of symptoms and those that we see in patients with inherited mitochondrial diseases, and that prompted us along our course of investigation.
So were these patients who had unknown mitochondrial disorders, or were they exposed to drugs that can cause mitochondrial mutations?
So these were patients who'd been successfully treated for HIV infection. The virus was under control. Effectively, they seemed to be in remission or almost cured, but they were developing late complications like neuropathy, myopathy, and sometimes ophthalmoplegia and encephalopathy. And it was these features which together made us think about mitochondrial disorders and led to the research that we carried out. And we took a combination of approaches using muscle histochemistry, biochemistry, and in addition some next generation sequencing to try and tease out the mechanism. And in a nutshell, it turns out that these patients have accumulated secondary mutations of mitochondrial DNA which are very similar to those that we see in healthy aging individuals. The difference here is that these are individuals in their 40s who've had HIV for 10, 20 years, successfully treated, but their mitochondrial DNA looked like that of an 80-year-old, implying there was some form of accelerated mitochondrial aging mediated at the DNA level.
And do you think this was due to the drugs that they were treated with, or do you think it's the effect of the HIV infection itself?
Well, it's actually difficult to tease that out, but the evidence from our study was that it was the people who'd been exposed to anti-HIV drugs, and particularly the nucleoside reverse transcriptase inhibitors, at some distant point in the past that were vulnerable to these changes. And in fact, the patients who had HIV infection who weren't treated with these drugs didn't show these changes at all.
What would you think the implications of your research will be on clinical practice going forward?
So the interesting thing is that these patients were developing these secondary mitochondrial changes 10, 15 years after stopping treatment. Now, if we think that these drugs have only been around for, 20 years, then this is perhaps the tip of the iceberg that we're starting to see in our clinics, and this may be a late toxic effect that's only just becoming manifest. And I know from discussing this with colleagues around the UK and also in Canada that there are patients with features very much like mitochondrial diseases in the HIV community who are just starting to emerge. So it's conceivable that we're just starting to see the beginnings of a tip of the iceberg, if you like.
So do you think in terms of therapies for these patients, is there any new avenues of research that might be helpful?
Well, I think the most important thing for us in the Western world is that actually these drugs, nucleoside reverse transcriptase inhibitors, are not being used to the same extent, and in fact the newer versions are less toxic to mitochondria. So I don't think that for the future this is going to be a problem in the Western world because we're not using these medications anymore. But actually, if you reflect on sub-Saharan Africa, where HIV is clearly a big problem, these cheap drugs that are off license are being used extensively and could lead to substantial problems there in the future.